Chapter 12, drugs, microbes, the elements of chemotherapy. Okay. So the principles of antimicrobial therapy, the goal of antimicrobial chemotherapy is to administer a drug to an infected person that destroys the infective agent without harming the host cells. That's the goal. Can we do that? No. Cells, there's always going to be side effects, things that we don't want. That's the goal, to kill the invading infectious agent and leave all the other human cells, quote, normal cells alone. Antimicrobial drugs are produced naturally or synthetically. We can make them. The perfect drug does not exist. Okay? But by balancing the drug against one another, a satisfactory compromise can usually be achieved. Characteristics of ideal antimicrobial drug. We want a selectively toxic to the microbe, but non-toxic to the host. We want it microbicidal rather than microbistatic. We want it to kill the bacteria. Remains potent long enough to act and is not broken down or excreted prematurely. Has to remain long enough. Is not subject to the development of antimicrobial agent resistance. Okay. And it complements or assists the additives of the host defense, their natural immunity. Remains active even when diluted in body fluids. Readily delivered, got to be able to get it where it's supposed to go, to the site of infection. And if we're talking about US medicine, it has to be reasonably priced. And does not disrupt the host's health by causing allergies or predisposing the host to other infections. Okay. With penicillin treatment, one of the big problems with penicillin is in females, yeast infections. That's one of the things that causes by it predisposes health to other host infections, like killing off the good bacteria along with the bad bacteria. Okay. Antibiotics. Okay, are common metabolic products of aerobic bacteria and fungi. Molds in generally the Penicillium and Cephalosporium. They act by inhibiting the other microbes in the same habitat. Antibiotic producers have less competition for nutrients in space. So it works in the same habitat that bacteria live in and depletes them of their nutrients in the space. You see how it affects the different colonies in the petri dish here. So penicillium is a mold, cephalosporin is a mold, Micromonospora is a bacteria, Bacillus is a bacteria, Chromobacterium is a bacteria, and Streptomyces is a bacteria, filamentous bacteria. And these are the drugs that form. Some of our major drugs are penicillin, cephalosporins, gentamicin, bacitracin we use all the time, streptomycin, erythromycin, tetracycline, vancomycin, etc. All right, so here's some different terms that we use. Chemotherapeutic drug, okay, is any chemical used in the treatment, relief, or prophylaxis of a disease. Prophylaxis means you're giving a drug to prevent an occurrence. People that come in for dental work, if they've had, say, knee surgery within 2 years, or a metal disc placed in their back or in their knee, we give them antibiotics an hour before their appointment. That's a prophylaxis. That's a preventative treatment to stop any bacteria from lodging on those parts and causing an infection on that foreign body that's been implanted in their body from surgery. Okay? It's a way of preventing an infection. Oh, prophylaxis, just like above, prevent potential infection and person risk. Antimicrobial chemotherapy, the use of chemotherapeutic drugs to control infections. Antimicrobials, used for any antimicrobial drug, regardless of its origin, something that kills bacteria. Antibiotics, produced by the natural metabolic process of microorganisms that can inhibit or destroy other microorganisms. Semi-synthetic drugs, drugs that are chemically modified in the laboratory after being isolated from a natural source. So they're semi-synthetic. Synthetic drugs, antimicrobial compounds synthesized entirely in the laboratory through chemical reactions. We make that through chemical means. Narrow spectrum means it's very limited. It only affects bacteria of a certain type, like it only works on gram-positive bacteria, only works on gram-negative bacteria. Broad spectrum works on a whole range of bacteria. So that's some of the terminology we use. Prophylaxis, to keep guard before, but in Latin, prevents infection in a person at risk. We don't give that to everyone. We only get people at risk. Chemotherapy is when we use a drug to treat a disease like cancer. Antibiotic goes against bios, against life of the microbial agent. So the antimicrobial drug should be selectively toxic. We should select the toxicity to only work on the microbes that we want. Is that possible? No, but we can control it to a certain extent. Drugs should kill or inhibit microbial cells without damaging the host tissues at the same time. As the characters of the infection agents become more similar to the vertebral host cell, vertebrate host cell, sorry, complete selective activity becomes more difficult, okay, and more side effects are seen. So the closer the infection agent is to the host cell, the more difficult it's going to be to separately select it out to kill that one, to kill that microbe before harming the body. There's 5 major components that are useful for drug targets in an actively dividing cell. You can inhibit inhibition of the cell wall synthesis. You can stop from making a cell wall. That can't mean the wall can't survive. Breakdown of the cell membrane. Okay, breaks down the cell membrane. Insides go outside the cell lysis and becomes non-functional. Interfere with the functions of DNA and RNA. You can't take DNA to RNA, you can't make your proteins. Inhibition of protein synthesis can affect the ribosomes. Stop your protein synthesis. Blockage of key metabolic pathways. Block any of the pathways that are required because DNA has to go to our messenger RNA. Messenger RNA goes to ribosomes. Ribosomes go to transfer RNA, and then they staple together to make the protein. So if you block any of those pathways, you're going to block the action of that microbe. These are different drug use and how they affect. We'll do a couple of these. Penicillins, cell wall damage and lysis, causes cell wall break. Cephalosporins, again, cell wall damage and lysis. Bacitracin, cell wall damage and lysis. The nice thing about these 3 drugs is it affects the cell wall. And what's nice is bacteria have a cell wall. Human cells do not. That's one of the nice effects of those drugs. Niacin we use a lot. Okay, that inhibits ribosomal protein synthesis. Vancomycin, cell wall damage and lysis. Tetracyclines inhibit protein synthesis on the ribosome itself when it tries to put the primary amino acid chain together by reading the messenger RNA and putting transfer RNA codons onto it. Sulfa drugs inhibit folic acid metabolism. What else do you want? Quinines were used. That's what originally one of the treatments for the COVID. Buildup of toxic waste in the parasitic cells, what it causes. Metronidazole, buildup of toxic free radicals. That's real common. The cyclovirs are used to treat viruses. It stops with viral replication. Azithromycin, this is used in treatment of AIDS and the blocks formation of DNA from RNA strands. Okay. So we're blocking different areas. You can block the cell wall without a fully functional wall. The cell's lysis. You can affect the cell membrane. Okay. Things can leach in and out. You can affect the DNA and RNA. Can't replicate it. Okay. You inhibit transcription. So you can't add the transfer RNA to the protein chain. You can't make your proteins. Interfering with the ribosomes, disrupt protein synthesis. And any metabolic pathway we can add to the enzyme that control them and alter its shape so they cannot be used. So again, antimicrobial drugs, we have the narrow spectrum and a medium or broad spectrum. Narrow spectrum is an affects only a small range of microbes. Targets a specific cell, not only in certain microbes, like bacitracin is narrow spectrum. Bacitracin is a drug you use it over the counter. It's in it's cuts, things like that. You put it on it, and helps prevent infections. It's like an ointment or cream. Broad spectrum drugs has a great range of activity. They target cell components common to most pathogens. They don't target a certain cell type or certain cell component. It's like ampicillin, tetracycline. Those are broad spectrum antibiotics. All right. Next question, or first question on this chapter. A drug that targets lipopolysaccharides in a gram negative would be considered broad spectrum, narrow spectrum, selective toxicity, broad spectrum and selective toxicity, narrow spectrum and selective toxicity. So it targets the lipopolysaccharides, so it's very in grams. So it selects a certain one and only targets the membranes where it has the lipids in there and it breaks those apart. So it's not either broad spectrum or narrow spectrum, but it's very selective. You don't know if it's broad spectrum because there can be a lot of gram-negative bacteria. So I would say it's very selective toxic. You don't know if it's broad or narrow spectrum from the definition yet. That's selective toxicity. Because it selects only those cells that works on that lipopolysaccharide membrane, and it's only in the gram-negative bacteria. So only affects gram-negative cells, and it uses a certain spot in that cell membrane. I mean, you could make an argument for narrow spectrum also. I would give you credit for that, narrow spectrum. The most correct answer is selective toxicity. That makes sense?
Yeah, like if it had like an actual like disease or infection, then it would be either narrow or broad, because then we could determine which one it would be.
Determine what it is, yeah. Yep, exactly. So antimicrobials that affect the bacterial cell wall. That's what we want, because human cells don't have cell walls. Most bacterial cell walls contain peptide glycans, penicillin, and cephalosporins block synthesis of peptide glycans, causing the cell wall to have holes in it and lyse break apart. It's active on young growing cells. Penicillins that do not penetrate the outer membrane and are less effective against gram-negative bacteria. Broad spectrum penicillins and cephalosporins can cross the cell walls of gram-negative bacteria and lyse their membranes. Cephalosporins are much more powerful antibiotic. If penicillin doesn't work, the next one of the choices is cephalosporins. Cephalosporins are used for urinary tract infections, for very large abscesses in the mouth. Okay. The only thing with penicillins is if you're allergic to penicillin, there's a 15% chance you're allergic to cephalosporins also. Something like Keflex, drugs like that. So be very careful when you're giving a patient that's allergic to penicillin or cephalosporin, because 15% of those will also be allergic to cephalosporins with penicillin allergies. Effects of penicillin on gram-positive cells wall. You can see it affects the cell wall, allows the cell, it breaks it down, allows the cell to lyse, to break apart. It gets in between the formation of that cell wall. You can see in the scan electron microscope how it puts holes in the cell wall and causes it to lyse. Okay, so normal, okay, the left and center views are Staphylococcus cells on that previous slide. So the left and center views here are your normal cells. Okay, the right view is the molecular structure of the peptide glycan, which is what penicillin attacks. Okay. If it's affected with penicillin, the left side shows a breakdown of Staphylococcus cell exposed to penicillin when it explodes. The center view shows the actual cell undergoing lysis and cell death. Right indicates how penicillin blocks the formation of the cross bridges that dismantle the framework that holds the cell all together by doing these peptide glycans. It stops the peptide glycans from linking inside the cell wall, leaving holes in it, and then the cell can leak out and lyse. So a cell with a damaged membrane dies from disruption in metabolism or lysis. These drugs have a specificity for a particular microbial group based on the different types of lipids in their cell membranes. Polymyxins interact with phospholipids and cause leakage, particularly in gram-negative bacteria. Amphotericin B and nystatin form complexes with sterols on the fungal membrane, and that causes lysis. Those are antifungal medications. Okay, and you see polymyxin gets incorporated into the cell membrane and it dislodges where it comes through between there, through the phospholipid layer, and causes openings in the cell membrane, breaking down. Okay, there's drugs that affect nucleic acid synthesis, making of DNA and RNA. These things like synthesis of the nucleotides. You can inhibit replication or stop transcription. Chloroquinone binds and cross-links a double helix. Quinolones inhibit DNA helicases. It doesn't allow the DNA to coil for replication. Okay. Antiviral drugs that are analogs of purines and pyrimidines insert into the viral nucleic acids, preventing the virus from replicating its DNA inside a cell. Okay. Drugs that block protein synthesis. Ribosomes of eukaryotic cells differs in their size and function from prokaryotes, okay, but they can also damage the eukaryotic mitochondria. Okay. Aminoglycosides like streptomycin and gentamicin insert on sites of the subunit, the 30S subunit of the ribosome, and cause misreading of the messenger RNA. Okay. So that it cannot replicate to its proteins correctly, and the cell dies. Tetracycline blocks attachments of transfer RNA on the A acceptor site and stops synthesis. Remember the polypeptide chain for the primary protein structure. Okay, messenger RNA comes into the subunit. The transfer RNA has 3 codons. They link to it and they add the amino acid. Link to it, add amino acids. Okay, this stops that transfer RNA from attaching the amino acids onto the peptide chain. So it disrupts, it stops the primary protein structure from forming. So here's your aminoglycoside. It bonds to the ribosome and it stops these transfer RNAs from bonding in here and transferring an amino acid to create this long amino acid chain as each one gets preceded. So messenger RNA is misread and the protein is incorrect and the cell dies. Chloramphenicol, formation of the peptide bond is blocked. It comes up here and it blocks the formation of the peptide bond. So you can't link amino acids together. Ozolidinones, okay, prevent initiation and block the ribosome's assembly. So these 2 units of ribosome, the large unit and small unit, can't come together. So you can't get that stapling effect that puts the transfer RNA amino acids together. Tetracyclines. Tetracycline blocks the transfer RNA from coming in here. So transfer RNA cannot get in with its amino acid attached to it. Okay, so it cannot get in here and bind to that and form the primary structure of that protein chain. Erythromycin, okay, prevented from translocating. It blocks the chain here and prevents the messenger RNA from advancing through the ribosome and reading each unit. It stops the advancement of the messenger RNA through the ribosome. So no protein can be formed. Competitive inhibition, which when a drug competes with the normal substrate for the enzyme's active site. So in other words, if you have a site like that, and you're going to get a fit, can fit right in there, right? If you get another drug that has the same site, same shape, it can bind in here. If you get more of this building up, it's going to compete with the substrate normal that goes in there. And it's going to be a competitive inhibition. It's going to bind it where the other drug can't. Okay, so metabolic antibodies are dead end and cannot function as required because it blocks the site. As the enzyme is no longer able to produce a needed product, cell metabolism slows down or stops. So sulfonamides, retrovir are examples of drugs that do that. Okay, that's what's called competitive inhibition. It competes with the binding site. Synergistic effect. The effects of a combination of antibiotics are greater than the sum of the effects of individual antibiotics. So sulfonamides and trimethoprim combined block enzymes required for tetrahydrofolate needed for DNA and RNA synthesis. So the two can work together to stop the synthesis of RNA or DNA and inhibit the microbe from growing or replicating. Competitive inhibition we talked about. Okay, so sulfonamide can inhibit the enzyme. So the metapod need to synthesize a tetrahydrofolic acid. contains two enzymes that are chemotherapeutic agent. So under normal circumstances, PABA acts as a substrate for the enzyme pteridine synthetase, and the product is needed for the eventual production of folic acid. So you can stop it at different metabolic sites. In other words, PBA goes to dihydropyric acid, dihydrofolic acid, tetrahydrofolic acid, and they produce amino acids, pyridines, and purines. Okay? So folic acid is required to synthesize that as a coenzyme. Okay? So these are the different steps involved in it. So if you inhibit any of those steps, it will block it. Okay? So we can have competitive inhibition. We should talk about this is the PABA, and what it normally does is binds to the site. Now you get something that has that same shape that bonds to it. This is a sulfa drug. If you put more of the sulfa drugs as it gets higher levels, it starts blocking this site, using it up. So the PBA can't be used. That's competitive inhibition. So since little or no PBA can bind now anymore, because the other sites being sealed, okay, the synthesis of folic acid will be inhibited. Okay. So aminoglycosides are antibacterial agents that target either peptidoglycan synthetase, the cell membrane, protein synthesis, or DNA replication. Let's go back to aminoglycosides.
Is it C?
I think you're right. Let's go back to aminoglycosides. Here's your aminoglycosides. What aminoglycosides do is bond to this ribosome, so proteins can't be made. Okay, so aminoglycosides. Okay, so the protein can't be made, they block protein synthesis. So it would be C. Exactly right. Good memory. Very good memory. Okay. There's 280 different antimicrobial drug classified in 24 drug families. Antibacterial drugs that act on the cell wall are penicillins and its relatives, cephalosporin group of drugs, carbophenamines and monobactrams, and miscellaneous cell wall inhibitors, non-beta lactam drugs. Antibodies that dominate bacterial cell membranes, and drugs that act on DNA and RNA. There's 3 different types right here that you can classify. Drugs that act on the cell wall, drugs that act on the cell membrane, or drugs that act on DNA or RNA. You know that drugs that interfere with protein synthesis, like your aminoglycosides we just talked about, tetracycline, chloramphenicol, macrolides, and related like that, or drugs that block metabolic pathways. We talked about folic acid synthesis. It can block any one of those steps and have the same effect of not producing folic acid, so you cannot use it as a coenzyme for protein synthesis. Okay, beta-lactam. Lactam is the ring of bacteria. So you have beta-lactam antimicrobials, all contain a highly reactive 3-carbon, 1-nitrogen ring. Primary mode of action is to interfere with cell wall synthesis. Okay. Greater than half of all antibiotic drugs are beta-lactams. They interfere with the cell membrane. Penicillins and cephalosporins are the most prominent beta-lactams. Okay. This is your beta-lactam ring. Okay. That's the structure of penicillin. Okay. Penicillin in the groups like ampicillin and amoxicillin has a whole bunch of cillins. Okay. Large diverse group of the compound can be synthesized in the laboratory. More economical to obtain natural penicillin through microbial fermentation than modify it to the semi-synthetic forms. But all consists of 3 parts. All the penicillins, or the penicillin families, its relatives, have a thiazolidine ring, have a beta-lactam ring, and there's a variable side chains that dictate this microbial activity. Penicillin G and V are the most important natural forms. It's a drug of choice for gram-positive cocci, streptococci, and some gram-negative bacteria, meningeal and syphilis. Semi-synthetic penicillin is ampicillin and amoxicillin, have a broader spectrum for gram-negative infections. Penicillin H resistance, methicillin, naphthalene, cloxycycline, and primary problems are allergies and resistant strains of bacteria. We use those so much, we're getting resistant strains of bacteria to these beta-lactams. And we're trying to develop a lot of patient allergies to it because we're using so much. Okay, so this is penicillin V, which stands stomach acids being taken by mouth. This is ticarcelin, extended spectrum, okay, effective against gram-negative rods. Floxacillin, broad spectrum, resistant to penicillinase. Nafcillin, narrow spectrum, okay, resist penicillinase. Okay, ampicillin, for the first penicillin to provide broad spectrum. And methicillin, first penicillin to resist the effects of penicillinase. Those all come from the main structure there. So cephalosporin, to talk about penicillin, we talk about cephalosporin. Cephalosporins account for 1/3 of all antibiotics administered. Synthetically altered beta-lactam structure. Okay, they're relatively broad spectrum, resistant to most penicillinases. So bacteria that can break down penicillins can't break down the cephalosporin. Causes fewer allergic reactions than penicillins. Okay, some are given orally, like Keflex. Keflex is your major one that we treat patients with. Some are given orally or can be administered principally also. Generic range are Cef, or Kef. Okay. These are new improved versions of the drug referred to as the new generations of cephalosporin. 4 generations exist. Each group is more effective against gram-negatives than one before, with improved dosing schedule and fewer side effects. So you have first, second, third, and fourth generation. Okay. So cefazolin, first generation, was most effective against gram-positive cocci and a few gram-negative. The second generation, more effective against gram-negative bacteria. Third generation, cephalexin, this is Keflex here, cephalexin. It's a broad spectrum activity against enteric bacteria with beta-lactamases. Beta-lactamases prevent beta-lactam rings from breaking down the cell walls. Cephalosporins bypass that. Okay, the bacteria is still sensitive to it. Cefepime is the fourth generation, is the widest range, gram-negative and gram-positive. Additional drugs we use to take that are beta-lactam drugs that break down the beta-lactam ring in bacteria. Carbapenems, okay, broad spectrum drug and infections with aerobic and anaerobic pathogens. Very low dose, administered orally with very few side effects. Monobactams, as you know, narrow spectrum drug for infections by gram-negative aerobic bacilli, may be used by people allergic to penicillin. Vancomycin, okay, very narrow spectrum, both in treatment of staphylococcal infections, in cases of penicillin and methicillin resistance, or for patients allergic to penicillin. It's toxic, hard to administer, so it has restricted use with vancomycin. Bacitracin, that's over the counter, has a narrow spectrum produced by strains of Bacillus used topically in ointment. And INH, isoniazid, okay, it's what we use to treat tuberculosis, mycobacterium tuberculosis. It interferes with the mycolic acid synthesis. Like I said, treats tuberculosis. Polymyxins, a narrow spectrum peptide antibiotic with a unique fatty acid component. It's indicated for drug resistant, Pseudomonas aeruginosa, and severe urinary tract infections. Fluoroquinolones bind to the DNA grayase and topoisomerase 4. It's a broad spectrum, is its effectiveness, and concerns arisen regarding the overuse of quinoline drugs. Quinoline is a very powerful antibiotic. We generally don't prescribe that often. It has some tremendous side effects. The Center for Disease Control recommends careful monitoring use. If you have a disease, a bug, like microbe, that is resistant to penicillin, erythromycin, methicillin, okay, we use quinoline. Quinoline is a nasty drug. I had it once and I was really sick. And I was in the hospital, in fact, I was in the hospital, the one in Yucca Valley. And this was in June. And I looked out the window and told them it was snowing. It really affects your mind. I was on this drug when they sent me home, this quinoline. And my wife found me in the backyard laying the pigs out of the backyard, opened the gate for them. And we don't have pigs. it produces vivid hallucinations.
Wow.
Yeah, it's a drug of last resort, but it works. Like nothing else is working. And I was getting sicker and sicker. So it does have some severe side effects. So it's saved for really severe infections and resistant to other forms of antibiotics. But yeah, because you get hallucinations that and they're real. They are real hallucinations. It's amazing. So, like I said, it's reserved, it's carefully monitored on the quinolines. Aminoglycosides, you talk about how it disrupts the protein synthesis. Product of various species of soil, Actinomycetes. It's a relatively broad spectrum. Okay, inhibits protein synthesis, bind to one ribosomal subunit. So it binds to one subunit and the units don't come together. Useful against aerobic gram-negative rods and certain gram-positive bacteria. Okay, streptomycin, that's for the bubonic plague, tularemia, and tuberculosis. Gentamicin, less toxic, used against gram-negative rods. And the newer one is tobramycin and amikacin, used against gram-negative bacteria also. Okay, these are some of the drugs that interfere with protein synthesis. Here you have a 6-carbon ring in the center there, and it's bonded to 2 different sugars. One 5-carbon, one 6. One 5-point, one 6-point. Okay. Tetracyclic antibiotics. Tetracyclic antibiotics are broad spectrum, block protein synthesis by binding to the ribosomes. Their treatment for STDs, Rocky Mouse spotted fever, Lyme disease, acne, and different protozoal infections. Generic tetracycline is low cost, but limited by its side effects. One side effect of tetracycline is on teeth. Okay. Teeth they'll come in, they'll develop, your tooth comes in, there's your tooth, kind of a block of a tooth there. You'll have a gray band where you were given that tetracycline as a child, and this will be gray. Okay. It's very difficult to bleach that out. It's tremendous aesthetic effect of tetracycline, called tetracycline standing in teeth. I think I have a picture of it in here too. So tetracyclines interfere with protein synthesis. They stop the ribosomal subunits from coming together, and they block messenger RNA translating with tRNA to make into a protein chain. Chloramphenicol also blocks protein synthesis. It's a potent broad spectrum drug with unique nitrobenzene structure. Our benzene ring blocks peptide bond formation and protein synthesis. Entirely synthesized through chemical processes. There's no natural component to it. It is very toxic. We have restricted use of it. It can cause irreversible damage to bone marrow. Bone marrow is where you make your red blood cells and your immune system, your B cells, your T cells. You don't want to damage that bone marrow. So it's very restricted use. It can cause some severe problems. Typhoid fever, brain abscesses, varicocele, and chlamydial infections. Okay, are used in treatment of chloramphenicals, are used to treat those. This is your benzene ring. This is your nitrogen group on it. Okay, that's your chloramphenicol. What's the chemical structure of it? Okay, macrolides and related antibiotics. Erythromycin. Erythromycin has a lactone ring with sugars attached to the ribosomal subunit again, ribosomal subunit. It's a broad spectrum, very low toxicity. Erythromycin you can use all the time. It's taken orally for different types of pneumonia, Legionnaire's disease, chlamydia, pertussis cough, diphtheria, and it's a prophylactic prior to intestinal surgery. Used to prevent infections. It's good for penicillin resistant people with gonorrhea, syphilis, acne. They treat with erythromycin if they're allergic to penicillin. Newer semi-synthetic ones are clarithromycin and azithromycin. This one is used, it is AZT, this is used in AIDS treatment. And this is what erythromycin is. Sugar groups attached to your main carbon rings. Okay. Like I said, different metabolic pathways are mostly synthetic. Sulfonamide, or sulfa drugs are the most important. Now, when you're working in the real world, a lot of patients have sulfa drug allergies. So you cannot use these sulfonamides because they've been around so long. A lot of people have penicillin allergies too, but sulfa drugs have a lot of allergies with patients. So you have to be very careful with that. They're narrow spectrum. They block the synthesis of folic acid that's produced by bacteria and needed by it. Okay. Sulfoxyl has produced for shingles, urinary tract infections, protozoal infections. Silver sulfadiazine for burns and eye infections. Trimethoprim, given in combination with other sulfa drugs for UTIs and PCPs. Okay. So this is the different groups attached. Here's the compound. This is your main group here. And where this bond is here, these are the attachment. You can attach this one, you can attach this one, you can attach this one. So different attachments to it for different sensitivities to different microbes. It's made synthetically. Newly formed class of antibiotics, formulated from a pre-existing drug class. There's 3 new drug types. Foxfomicin, trimethamine, is a phosphoric acid derivative, alternate treatment for UTIs, and it also inhibits cell wall synthesis. Cindercid, effects against Staphylococcus and Enterococcus that cause endocarditis and surgery infected. To use bacteria resistant to other drugs, it inhibits protein synthesis. Endocarditis can be a fatal infection. Endocarditis is an effect we talked about a little bit before that law case I had. The heart pumps blood and the valves open and close so blood can flow through the heart. If there's a murmur or something backs up the blood of the heart, bacteria in the bloodstream can lodge on that valve. That's what endocarditis is. It's an infection on the heart valve itself, which can be fatal. Daptomycin is one of the new ones too, directly made against gram-positive bacteria, disrupts the membrane functions. Okay, drugs have other ones that have metabolic pathways, ketolides, Ketec, different ring structure from erythromycin, used for respiratory tract infections and resistant to macrolides. Okay. Zyvox, synthetic antimicrobial that blocks interaction with the messenger RNA on the ribosome. So you get your 2 subunits of your ribosome coming together, and your messenger RNA, it blocks this messenger RNA from coming to the ribosome, so you can't make proteins. Used to treat methicillin-resistant Staphylococcus aureus. Those are your MRSA infections. Remember how dangerous MRSA was because nothing was treating it? This is the new drug that treats it. Okay, and vancomycin-resistant Enterococcus. So MRSA and VRE. All right, which of the following drugs inhibits bacteria by blocking folic acid synthesis? Which one of those blocked folic acid synthesis?
Is it, I'm just guessing at this point, is it A?
I would say A,
Well,
if I remember right. No, it's not. It's sulfonamides. I was trying to remember what they were saying. It's sulfonamides. The sulfonamides, that's right. The sulfonamides block folic acid synthesis in bacteria. Chloramphenicol was, where was I thinking? Sulfonamides block the synthesis. Yeah, sulfonamides, narrow spectrum block the synthesis of folic acid. Those are your sulfonamides. Chloramphenicol blocks the formation of protein synthesis, not folic acid. It's not a metabolic pathway. Okay, the sulfonamides block the metabolic pathway. I got it mixed up. All right, so it's C, sulfonamides. The block the synthesis of folic acid is needed by the bacteria. Now, antifungal drugs. We talked about antibacterial drugs. These are antifungal drugs, destroy fungal infections. So the cells of fungi are eukaryotic. They present special problems in chemotherapy because the fungal cells are eukaryotic. They don't have a cell wall. So our cells are eukaryotic, so the cells are similar. So now we got another problem. We've got to find a way to kill cells similar to ours and not kill our cells. So the great majority of chemotherapy agents are designed to act on bacteria because bacteria have cell walls and we don't. Okay? They are generally ineffective in combating fungal infections. Fungal cells are eukaryotic. A drug that is toxic to fungal cells is also capable of harming human cells. They're both eukaryotic cells. There's 5 main groups of drugs that have been developed to specifically treat fungal infections. Macrolide, polyenes. They bind to the fungal membranes, cause loss of their selective permeability. Amphotericin B and nystatin are the 2 most common drugs. Amphotericin B is the most versatile and effective antifungals. It's a topical. It's also a systemic. You can take it. Nystatin is a rinse. We use it with a lot of our denture patients. It's only a topical treatment. Okay. Grisofluven is stubborn cases of dermatite infections. It's nephrotoxic, toxic to your liver, your kidneys. Sorry, toxic to the kidneys. Synthetic azoles are broad spectrum, different types of medications there. Fluticin is an analog of cysteine. Okay, amphotericin B for systematic mycosis, and sulfocandins damage cell walls for capsule fungus. Okay, so those are your 5 different groups. Again, they're different types of bacteria. Remember, they don't have they're eukaryotic cells. They don't have the cell wall. This is like a combination with bacteria too, is with it also. Okay, these are your antifungal drugs. That's what they look like. Long chain fatty acids here. Ring structure similar to the benzene ring. Okay. Antiparasitic chemistry. Antimalarial drugs, quinine, chloroquine, primaquine, and mefloquine. Antiprotozoan drugs, flagyl, okay, tetracyclines, sulfonamides, antihelminth, I can't say that word, antihelminth drugs, okay, immobilize, disintegrate, or inhibit metabolism. Okay, mebendazole is a broad spectrum, inhibits the functions of microtubules, interferes with glucose utilization, and then it disables them. Okay, it paralyzes muscles, pyrantel, and piperazine. Okay, niclosinamide destroys scolex. Those are for different parasites. Antiviral chemotherapy agents. Okay, selective toxicity almost impossible due to the obligate intercellular parasitic nature of viruses. The viruses inhabit a normal human cell or host cell, so it's impossible to tell the difference. So it's very difficult to treat. Then most of these are designed to block a step of the virus cycle. Some of the cells block the penetration of the virus into the host cell. Some block the replication, transcription, or translation of genetic material once it's in the host cell. Acyclovir, it's one of those common ones we prescribe. Herpes virus on your lips, inside the mouth. If you apply it to the side of your lips when you first notice it, it helps decrease the length and duration that you have the herpes lesion on your lips. Ribavirin is a guanine analog. It's a reverse synthetic virus, hemorrhagic fevers to treat. AZT is used in treating AIDS, and it's a thymine analog. Or we can prevent the maturation of the virus particles. Protease inhibitors. Proteases put the proteins together. This blocks the proteases from working. Okay, antiviral chemotherapy agents. Drug for influenza. Influenza is the common flu. Amotidine, Rimetine, essentially discontinued as flu treatments. Currently, viral strains have developed almost complete resistance to them, so we don't use those anymore. That's why the flu vaccine is redeveloped every year. Relenza and Tamiflu, slightly broader spectrums, blocks neuraminidase in influenza A and influenza B. So we can inhibit drugs by their entry or release of the cell. By Fusion, it's an anti-HIV drug, prevents binding of the viral particle, okay, to the cells, okay, and blocks the fusion of the virus to the cells. So when the cell has to, this is your virus that comes down here, the RNA coming down, and here's your cell. So these receptors that the virus binds to, it blocks these receptors, so the virus cannot bind to the cell. Maraviroc covers up the receptors, and HIV cannot adhere to remain, and it remains inactive. Same principle. Okay, it blocks the receptors. It binds to the receptors, so HIV virus cannot bind to it. Okay, Tamiflu. Okay, drugs do not stop the influenza cycle involving an entry, uncoating, replication. Okay, the virus enters, uncoats, replicates. They block the action of neuraminidase that are required for the final budding and release the new viruses. So the viruses are made in the cell, but now they can't be released. So this is the drug that blocks the receptor site. Okay, it binds to the cell. That's number 1 we talked about. That was this the fusing inhibitors of virus entry. Okay, so this stops virus entry. These little pink things right here block or bind to the receptors. Okay, the second one is the receptors get filled with a different material, so this cannot bind to it. Very similar in action. Both the first 1 and 2 prevent the virus particle from binding to the cell. The third one, like we said, the virus binds to the cell, goes into the cell, goes to the nucleus, makes the viral particles, assembles them, and goes to leave it. And then right here, these proteins here stop the virus from leaving the cell. It doesn't allow the cell to release the mature virus particles that have been made. Anti-herpes drugs, many antiviral drugs mimic the structure of nucleotides and compete for the sites on replicating DNA, like acyclovir. Okay. Oral and topical treatments for oral and genital herpes, chickenpox, and shingles. Okay. Inhibitions in nucleic acid synthesis. We stop the synthesis of RNA and DNA. Okay. This is cycloverse. So the nucleotide analogs that terminate DNA replication in the herpes virus synthesis. Okay. So it stops the DNA from replicating. Nucleoside analog reverse transcriptase inhibitors. This stops the action of reverse transcriptase, blocking viral DNA synthesis. Remember, the viral has the DNA component, and then it goes to RNA, and it makes the proteins. Right? It stops, okay, reversing the RNA back to DNA, so it cannot control the mechanism of the cell. Okay, remember, for these viruses, it injects the virus into the cell as RNA, and this RNA is converted into DNA. The DNA then goes into the nucleus of the cell, and it replicates. This stops RNA going to DNA, reverse transcriptase of it. Non-nucleoside reverse transcriptase inhibitors. Nevirapine attaches to the HIV RT binding site, and it stops its action of allowing the nucleotides to function to replicate the viral particles. Okay. So here's your stoppage point of it. Here, it stops the RNA from going into the DNA, incorporating into the DNA. So it stops this process right here. It doesn't allow the RNA to transcribe into DNA from the virus. Okay. So there's no reverse transcription here. It binds to this. It doesn't allow the bacteria, the virus, to get into the cell. Okay. Drugs that treat HIV infections and AIDS. There's a lot of drugs that can treat in combinations to manage HIV. Okay. All of the antiviral drugs target a specific part of the multiplication cycle of the human immunodeficiency virus. There are currently drugs that inhibit or block every strategic step in the virus cycle. So whether it's preventing from RNA to DNA, whether it's preventing from leaving the cell, whether it's entering the cell, we have drugs that work on all of those levels to try to stop this infection. The one here, antiviral chemotherapeutic agents are interferons. Interferons are human-based glycoproteins produced by fibroblasts and leukocytes. Therapeutic benefits include reducing healing time and some complicated infection, preventing or reducing symptoms of a cold or papillomavirus, slowing the progression of certain cancers like leukemias and lymphomas. Okay. And treatment of hepatitis C, genital warts, Kaposi sarcoma in AIDS patients. In AIDS patients, we see these black circles. They're little black lesions, or they can be very large, and they're very painful. And they're distinctive for AIDS patients. If you have a patient with their mouth, you see these black circles that are ulcerated on top of it. Those are Kaposi sarcomas. Those are an indication of the patient has AIDS. Okay, inhibition of HIV insertion, assembly, or release. Okay, it acts in different actions. Integrase inhibitor. This interactions with integrase and blocks actions HIV DNA is not spliced into the host chromosome. So this stops the DNA from the virus to getting into the host chromosomes and allowing that gene to multiply and be used. Protease inhibitors. Okay, these insert onto the HIV protease, enzyme that viral proteins turn viral proteins into functional pieces. Viruses are then defective and unable to find infected cells. Okay. So it stops the virus from maturing, leaves the cell to not mature viruses. They can't bind to other cells, so it basically inhibits the virus. Okay. So this is your first one here. It shows your virus goes from RNA to DNA. DNA enters the nucleus, but it does not allow it to get into the chromosome. So it can't be read to express its gene. The other one is the HIV protease. The HIV comes into the cell, but these drugs bind to it and prevent the HIV for the virus to mature. Okay, so it cannot kick out a mature virus. So the new virus cannot affect another cell because it doesn't have a mature virus. All right, so this is an overview we just talked about with binding inhibitors, fusion inhibitors, nucleotide reverse transcriptase inhibitors, nucleotide reverse transcriptase inhibitors, non-nucleotide reverse transcriptase inhibitors, integrase inhibitors, protease inhibitors, and fusion inhibitors. We just talked about. This is the acquisition of drug resistance. So drug resistance is an adaptive response, okay, in which microorganisms begin to tolerate an amount of drug that would ordinarily be inhibitory. So in other words, they get resistance to something you would normally be affected by it, but you've exposed to it so much that you've either evolved or developed resistance to it, so it doesn't infect you anymore. Same with microbes. So as a result of genetic versatility, okay, and adaptability of microbial populations, just like we adapt to things, can be intrinsic or can be acquired. Intrinsic means it comes from within. Okay, you're basically born with that response, or acquired, it's a learned response. Acquisition of resistance is the main problem for microbial chemotherapy, antibiotics. This is antibiotic resistance, if you're getting super infections because of this resistance that microbes have. Okay, so nuclei acquired after spontaneous mutations in critical chromosomal genes. In other words, genes that code for certain proteins, okay, they mutate and they can code differently now and they can kill off antibiotics. Acquisition of new genes or sets of genes by transfer from another species, like through the plasmid. You can transfer a section of DNA from a species back into another microbe. Okay, intermicrobial transfer, you transfer resistance factor called your R factor, okay, through plasmids. Plasmids are those small circular DNAs that can be incorporated into chromosomes. They're encoded with drug resistance. Okay, transposons duplicated and inserted from one plasmid to another, or from a plasmid to the chromosome. Give me ways of transferring that that form of DNA that has a resistance factor on it into the microbe, into the organism. And it shows you plasmid can transfer into, become incorporated into another plasmid, can become incorporated into its DNA, can be transferred from a virus going through its DNA, or can transfer from a DNA fragment of another organism back into the host organism. Right. Mechanisms of drug resistance, you can get drug inactivation. Okay. So this is penicillin in its active form. If the bacteria produces something called penicillinase, penicillinase breaks down penicillin or alters it so it's not active anymore. Okay. And you see the difference in penicillin here. You've added 2 methyl groups right there, and you've changed the ketone here to a carboxylic acid, and you opened up this bond right here. There's no more bond there to make a 4 cylindrical ring. So an activation of drug penicillin by penicillinase, okay, an enzyme that cleaves the portion of the molecule and renders it inactive. Okay, that's how drug inactivation works. You inactivate the antibiotic. Or decreased permeability. Okay, the receptor that transports the drug is altered. Okay, so the drug cannot enter the cell. Normally, the drug finds a receptor and is put in the cell where it can form its actions and cause the drug and kill the drug, kill the microbe. Okay, if you alter the surface, see how the difference, this is shaped like this, and this is shaped like this. So you alter the shape on the surface of those receptors, the drug cannot bind to the receptor and get pushed into the cell anymore. That's another way of resistance. That's called decreased permeability. Activation of drug pumps. Okay, normally the drug gets pumped into the cell, and you have it here where it can do its effect. Okay, a specialized membrane protein can be activated and pump the drug out of the cell. So it takes, once it's in there, this drug pump can pump the drug back out so it can't take effect. It's another way for resistance to occur. Or change in the drug binding site. Okay, here you got a triangle shaped antibiotic. It binds to that ribosome there, and it stops the ribosome from copying the messenger RNA and making the protein inside the microbe. Okay, if you change that shape, so you notice the shape here is changed to a circle now, not a triangle, so that antibiotic or that drug cannot bind there, and now the drug goes through and there's no effect on the ribosome, so it can make the protein that the to keep the cell alive, keep the organism functioning. Okay. So it's a basically binding site on the target ribosome is altered, so the drug has no effect. It can't bind, so there's no effect on that ribosome. Alternate pathway, alternate metabolic pathway. So this is the normal pathway the drug takes. We use the antibiotic right here. It blocks B going to C. So instead of doing that, it just takes another pathway. It goes from B to C1 to D1, back to the product. So it just switches, it takes a different pathway. So now the drug doesn't work because it doesn't block this alternate pathway. So basically the drugs have blocked the usual metabolic pathway, which is the top line, and then microbe circumvents it. We use an alternate pathway. Then that's an unblocked pathway, and it still sees the same outcome. All right. So which of the following effects do antiviral drugs not have? Antiviral drugs, they either kill extracellular viruses. Those are viruses outside the cell that haven't been infected yet. They can stop virus synthesis, which is an enzyme, inhibit virus maturation, okay, or block the virus receptors. So these three stop the virus from working. This one up here is just, they haven't even touched the cell yet. So there is no way it's going to know if the virus is going to bind to the cell. So the one that it cannot do or doesn't have for an antiviral drug is to kill extracellular viruses. Viruses haven't attached to it yet. We can stop its synthesis, we can stop it maturing, and we can stop the receptors from binding to it. But out in the open, we don't have any idea what it's doing. Okay? So it's killing extracellular viruses. Antiviral drugs do not have that effect. All right, natural selection and drug resistance. We talk about drug resistance, and we get it through natural selection. Large populations likely to include drug-resistant cells due to prior mutations or transfers of plasma we talked about. No growth advantage until exposed to the drug. In other words, there's a mutation, and this new bacteria is resistant to a certain drug because there's a mutation on the gene. Okay? There's no big advantage until that drug's introduced. You know, when the drug's introduced, it kills off all the others that are sensitive and leaves only those ones that are not sensitive. And then those multiply. That's where we get super infections from. So if exposed, sensitive cells are inhibited or destroyed, while the resistant cells survive. And then they start to grow. That's your advantage right here. And they start once the antibiotic is exposed to the microbe. Eventually, the population will be, the whole population will be resistant because you naturally select those that are resistant by killing off the ones that aren't. Okay. So here's your microbes. Okay, normal living a happy life. Okay, these are your drug resistant one, and there's no advantage to being drug resistant unless the drug's there. Now, we put in the antibiotic, start killing the infection off. It kills off all these drugs except for these. So these start to proliferate and make more and more of these. So eventually you get a colony of all microbes that are now resistant to the antibiotic that you use. That's what's causing our super infections. That's what's causing our problems. We are selecting bacteria by the unabated use of antibiotics, okay? And we're selecting bacteria that are resistant to those antibiotics. So now we get infections, these bacteria are now resistant to the antibiotics that we use. Okay? Microbial cells invade an infection. The most sensitive cells are eliminated by the drug. The resistant cells survive, natural selection, and then they begin to grow. So new cells that grow are resistant, and the infection will no longer respond to the drugs. That's the problem of natural selection. So physicians have the responsibility for making an accurate diagnosis and prescribing the right drug therapy. You need to know what sensitivity your microbes are to provide the right therapy for it. It does you no good to use the wrong antibiotic, because all that's going to do is select for bacteria that are resistant to that antibiotic, and it's not the right one in the first place. So you have to make sure it's the right antibiotic. You have to do testing. Compliant to the physician's guideline, the patient should take the correct dosage, the best route of administration, and for the right amount of time. This diminishes the selection of mutants that can resist low drug levels and ensure elimination of the pathogen.
Professor, so once the microbes go into the infection, so basically all the sensitive ones get erased or eliminated, and then the other ones pretty much start to grow. The ones that are resistant.
Correct, because they're not killed off. They're resistant to it.
So then they start multiplying and creating colonies then?
Right. Now you got a whole colony, a whole group out here that are resistant to that antibiotic.
Okay, got it.
And that's you pass on. Yeah, exactly. Combined therapy with 2 or more drugs together, okay, increase the chance that at least one of the drugs will be effective, and that a strain resistant to one drug will be eliminated by the other. So there are some indications we're using 2 antibiotics rather than 1. Research focused on developing shorter term, higher dose like antimicrobials. That's more effective, less expensive, and have fewer side effects. So you want a higher dose, kill them off real quick, doesn't give them the time to mutate and become resistant. Pharmaceutical companies continue to seek new antimicrobial drugs with structures that are not readily inactivated by microbial enzymes or drugs, with modags that are not readily circumvented. So we're trying to get drugs that these antibacterial actions are not sensitive, so they're not broken down, or big bacteria aren't going to evolve to be resistant to them. Our long-term strategy now are to reduce the abuse of antibiotics. Okay, we abuse antibiotics all the time. People come in with a sore arm or a headache or something like that. Antibiotics are not analgesics. Antibiotics do not fight pain. They shouldn't be given for pain. They should be given when a specific infection exists, and we know, or we're fairly certain, that an antibiotic will work under these conditions for that type of microbe. Okay, educational programs for medical workers, justification for prescribing certain types of antibiotics. Okay, valuable antimicrobes may be restricted in their use to only 1 or 2 types of infection. So in other words, we don't just dump all these bacteria, all these antibiotics on everything that comes through the door. We save certain antibiotics for certain types of infections so we don't develop these resistant strains. The additions of antimicrobials to animal feed, that's a big problem. Okay? Animals get sick, they die off, farmers lose money. So they put antibiotics in their feed to keep the cattle healthy. What that's doing is taking all the bacteria in that animal and creating bacteria that are resistant to antibiotics, and that transfers to human population. So now we got bacteria that are resistant to antibiotics because we fed them all these antibiotics to the animals. So trying to curtail that worldwide. It's hard to do when people are making money off it, though. Government programs make effective therapy available to low-income populations, so the right antibiotics are used, not just the cheapest ones. And vaccines should be used to help prevent the infection in the first place. So we're not giving all these antibiotics out. So those are our long-term strategies to help with this problem we have with super infections. Let's make that 5% of all persons with antibiotics will experience a serious adverse reaction to the drug, which is a side effect. No matter what drug you give, it will have a side effect. If any drug rep tells you that this drug has no side effects, they're wrong. Anything you give is going to have a side effect. It might be mild, might be severe, but it's going to have a side effect. Direct damage to major side effects, direct damage to tissue, toxicity of a drug. Okay. Allergic reactions, it can be an anaphylactic shock is an allergic reaction. Okay. Those can be severe. Disruption of the balance of normal flora. We kill off, remember you got good bacteria that live in your body too. It can kill off the good bacteria. And if you kill off the good bacteria, now the bad bacteria has more of an advantage, a niche where their food supply is more and they can spread out. And this helps to cause super infections. Okay. Gives a chance for these resistant bacteria to now overcome the space where the healthy bacteria we used to be living. So there are side effects to drugs. They have to be considered. Drugs, this is what's called tetracycline stain on teeth. You see this brown stain right here. Okay, that's tetracycline causes that. Again, when the teeth were developing in a child, you get that tetracycline. Very, very hard to treat. You can bleach it, but it's very hard to do. Okay, so basically what we're talking about when you have drugs in the your super infections, here's our drugs, okay, in the intestine. We give the antibiotic, okay, and it starts to destroy the beneficial flora. These red ones are the good ones. So all the good bacteria are being destroyed, and this leaves a whole lot of opportunity for these pathogens to overgrow now, causing super infection inside your intestines. Okay, these are some of this is a chart of some of the antimicrobial drugs and their the damage what they cause. Penicillin causes skin allergies. Ampicillin, diarrhea and enterocolitis. Cephalosporins, Keflex, decreased circulation to nephritis in kidneys. Okay, tetracyclines, discoloration of tooth enamel, diarrhea, tetracycline verifies for diarrhea. Become photosensitive, reaction to sunlight, chloramphenicol, injury to your white and red blood cells, precursors making red blood cells, aminoglycosides like streptomycin and gentamicin, diarrhea, enterocolitis, loss of hearing and dizziness. Okay, dizziness go along with loss of hearing, your inner ear for balancing your body with vertigo and kidney damage. One of the biggest problems of gentamicin, especially in young adults, is hearing loss, and that can be permanent. Okay, azithromycin is heart arrhythmias. Okay. Some of the other ones, let's see, some of the common ones, sulfonamide, sulfur drugs, blockage of urine flow, hemolysis, blockage of red blood cells, reduction in number of platelets. Okay. Let's see, some common antifungals, amphotericin B, disruption of kidney filtration, flucodisine, decreased number of white blood cells. Okay, metronidazole, nausea and vomiting, chloroquine, vomiting, headaches, okay, itching. The common drugs, acyclovir, common antiviral drug, can cause seizures, confusion, skin rash. Okay, amantadine, nervousness, lightheadedness, and nausea. Okay, AZT, this is a drug we use for AIDS treatment, immunosuppression, anemia. Okay, protease inhibitors, one of the causes can cause diabetes in patients. Okay. So just got to remember that all drugs, I don't care what it is going to have a side effect. Some can be severe, some can be very mild, but you are going to have side effects when you're giving somebody something. That's something you need to consider as a nurse or practitioner.
Yeah, I think that antibiotics are given out way too often and like abused.
Oh, definitely. The big problem is patients expect you to give them an antibiotic. Even if they have a cold, if someone has a viral infection, antibiotics do no good whatsoever. You never give antibiotics for the flu. But if a patient comes in your office, they want an antibiotic. They expect it. Well, you're not going to give me anything. Why am I here? You know, that kind of attitude. If you give medication, oh, you're giving me something. It's worth the money I'm paying now. We got to stop that mentality. We got to make sure patients, we got to teach or educate patients to understand that not everything is treated with an antibiotic. Certain diseases are just rest and fluids. You know, certain things you don't need prescription drugs. But like I said, if patients, you know, you're going to pay $300 for an office visit, you want to get a prescription to take home. You want to have something for you want to have value for your money. And that's the problem we're running into. Okay, because antibiotics do no good for pain. They do no good for viral infections. They can't treat the flu. If you have a bacterial infection and you know what bacteria it likely is, then antibiotics should be administered. But you will see antibiotic abuse all through clinics, and especially if the patient is very boisterous or very loud. I want something, I deserve it. And the doctor, the physician goes, okay, and gives it to him. And you've probably seen examples like that all the time. Okay. So selecting an antibiotic, at least three fashions: the nature of the microorganism that's causing the infection, whether it's gram-negative, gram-positive, whether it's an aerobe, whether it's an anaerobe. You're going to prescribe the correct antibiotic that's going to fight that infection. So specimens should be taken before antimicrobials are initiated. Now, that's not always feasible. A lot of times, you know, like with the oral infections in the mouth, I know penicillin is going to cure 90% of them. I know it's a gram-positive bacteria. I know it's an aerobe bacteria. I know it's going to treat it. Okay, so you treat it like that. Then if it doesn't work, then you take your specimen, but you get it started first a lot of the times. The degree of susceptibility, how sensitive it is to the drugs. Okay, the sensitive produced bacteria commonly showing you do different tests that will show if the bacteria is resistant to certain types of antibiotics. You can do a Kirby-Bauer disc diffusion test, an E-test, or a tube dilution test. Okay, tube dilution test uses different viscosities that you look at and determines the smallest concentration of drug you can use. This is called the minimum inhibitory concentration. That's the smallest concentration of the drug that will cause the desired effect. because why not use that? Okay, why give someone too much of a drug they don't need it? So if you know the minimum inhibitory concentration through this dilution test, you can see where it stops at what concentration. That's the concentration you give your patient. And the other one is the overall medical condition of the patient. If the patient has AIDS, whole different story of giving antibiotics. If the patient has diabetes, if the patient has congestive heart failure, patient has liver disease, kidney disease, those all affect how certain drugs metabolize in your system. You have to know your patient. You have to know your patient. You have to treat a patient, not a condition. That's what it boils down to. Yeah, this is a Kirby-Bauer disc test. You take a disc with the antibiotic on it, and you see how much of the microbe has been destroyed. And you can see these large circles around it show you how much of the construct you use and how much of this microbe in here has been destroyed around the disc that has the antibiotic on it. And you want to use the one that has the largest area, the largest circle of destruction. That's the most effective one. Okay. So it's evaluation of an aggregate fusion sensitivity test. This is the Kirby-Bauer disc that we just looked at. If the test bacterium is sensitive to drug, you get a zone of inhibition develops around the disc. That's that zone. Here's the disc you put in here, and then the zone of inhibition, and then the rest of the bacteria is all around it here. Okay. The larger the size of the zone of inhibition, the greater the bacteria's sensitivity to the drug. So the diameter of each zone is measured, evaluated for resistance, and compared to a standard. Then you make an educated decision of which antibiotic to use. Okay. You measure it, because this is the zone of inhibition. Here's all the bacteria. Here's the disc you put in, and this here is your zone of inhibition. How much of it does not have any bacteria in that region now? Okay, same thing. You can see the disc here, how there's nothing right here, but this one has a huge amount where the bacteria have been killed around. The bacteria has been killed around from the zone of inhibition. Right? Again, you can see the bacteria being wiped out in a particular zone. This is the dilution test, and you can see going from 1.6, from 0.8, you can see cloudy, 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 and then clear. It's clear like that. So you know the minimum dose would be at 0.8, between 0.8 to 1.6. You wouldn't want to give the patient 0.1, and you wouldn't get to 6.4. You want to give them the correct dose to kill the minimum dose you can use to kill the bacteria. Again, dilution tests you use in different wells, like different arrays that we saw before. Okay. And this is the minimum amount of the drug that you can use for the different types of bacteria. We've done different tests like Staph aureus, that's for penicillin G, ampicillin, and all the different ones. You can see how the different zones, okay, are going to be created, different translucencies. So the vitro activity is not always played with the vivo activity. In other words, what we do in the lab does not always match what happens in the human body. It's a guideline. We still have to treat our patient. If the patient's not responding, we can up the doses or we can switch medication. So just because it does something in the lab doesn't transfer 100% to a human body. It gives us a starting point, gives us an educated starting point, but you still have to monitor your patient. You never put a patient on a drug and send them home. I mean, you send them home, but you follow up. You make sure you don't just give someone a drug and say, oh, okay, take this and you're all better, don't call me again. I mean, you let them know if they develop a rash, call. If they develop difficulty breathing, call. If they're still getting sicker, call. And we switch it, we do things. Because not everything transfers exactly from lab to patient. You have a good percentage, but not all the time. So if therapy fails, a different drug, a different combination of drugs, or different administration might be considered. Might go to IV administration rather than oral. Okay. It's best to choose a drug with the highest level of selectivity, but the lowest level of toxicity. But you want to choose a drug that has the highest level of killing bacteria, the lowest level of killing our cells, toxic level for our cells, the host cells. And that's measured by what's called the therapeutic index. Therapeutic index is the ratio of the dose of the drug that's toxic to humans as compared to its minimum effective dose. You want a high index, you want a high index if possible. You want to have a high killing rate and low infection, low toxicity rate to humans. Okay, so in a tube dilution test, the MIC is blank of a drug that is required to inhibit growth of the microbe.
The smallest.
You got it. It is the smallest concentration.